I apologize. So Debbie Bangasser of Georgia State University was going to moderate this session, but she's her internet has been affected by the hurricane. So I am going to moderate this session, and just to save time, I'm going to introduce our first speaker, Dr. Elvisha Dhamala from the Feinstein Institute for Medical Research.
Hi, can everyone hear me?
Yes.
Okay, awesome. Let me just try to share my screen.
Looks good.
Okay. Perfect. Does that work?
Yes.
Great. So hi, everyone. I'm Elvisha Dhamala, and I'm an assistant professor at the Feinstein Institutes for Medical Research. And I'm super excited to be here because this is such an important topic. So thank you so much to the organizers for putting this together and also for inviting me to present here. So I'm going to broadly be talking about sex differences in brain and behavior. And since we already covered a little bit about what sex and gender are and aren't, I'm not going to go into too much detail there, but I do want to mention that sex and gender influence both our brain and our behavior. And it's important to recognize that this is happening throughout the lifespan, and this influences both our health and our vulnerability to disease. But we also need to recognize that sex and gender are not the same, but it is at the same time, they are both influenced by complex multidimensional factors, which includes a host of biological, social, and environmental factors. And a lot of times when we're actually studying sex differences, particularly in humans, we're actually capturing differences that may be driven by not just sex-related factors, but also gender-related factors. So we could be capturing things relating to hormones, chromosomes, the HPA axis, societal norms, gender identity, stereotypes, and individual experiences. So even though we want to focus on sex differences, we might inadvertently actually also be looking at gender. And another important thing to recognize is that different does not mean inferior. Throughout history, a lot of evidence that pointed to sex differences has actually been used to justify concepts of biological superiority or inferiority. But it's important to recognize that different simply just means different. It doesn't mean that one is better than the other. So with that being said, when we start to talk about sex differences in brain and behavior, there are a lot of differences in the prevalence of neurodevelopmental disorders, and this includes things like autism spectrum, ADHD, conduct, and intellectual disability. There's also differences in the presence of neuropsychiatric disorders, and this includes a whole range of them, including substance use disorders and addiction. And there's also sex differences in the prevalence of neurodegenerative disorders. And all of these are different disorders related to the brain, so obviously the brain plays a role. Across these classes, we need to recognize that we have these white matter fibers in the brain that directly and indirectly connect different brain regions, and these structural connections facilitate the flow of functional activation signals or neural activation signals between different brain regions. These structural and functional properties of the brain underlie our unique and individual behaviors and are also what tend to be disrupted and lead to different brain disorders, including things like addiction. Across the sexes, there are fundamental differences in both brain function and structure. In terms of brain structure, males tend to have about 10% larger brain volume than females, and this is consistent across gray matter, white matter, subcortical gray matter, as well as ventricular volume. In terms of brain function, there are notable differences in the types of connections that are more prominent in males versus females. So males tend to have more between module connections, whereas females tend to have more within module connections. And the topography or the organization patterns of the brain in terms of brain function can actually also be used to accurately distinguish individuals across the sexes. And this basically tells us that using information about different brain networks, and specifically what we call heteromodal or association networks, so networks like the ventral attention, default mode, frontoparietal, and the dorsal attention, we can actually distinguish across males and females. So taken together, this kind of suggests that there are these differences in the brain, and these differences in the brain may, in part, explain the differences that we see in the expression and prevalence of brain disorders and related behaviors. But not all sex differences are the same. A fundamental assumption of neuroscience research is that there is some relationship between brain and behavior that reflects the association or the relationship and the mechanism of influence from one to the other. When considering how sex differences in brain and behavior may impact us, it's important to recognize that these differences can exist in many different forms. We can have these differences in the brain, we can have these differences in the behavior, or in the relationship between the two. A lot of studies have focused on either sex differences in brain or sex differences in behavior, but in my lab, we actually study how sex influences the brain, the behavior, and the influences or the relationships between brain and behavior. So I'm going to briefly talk about the method that we use because it's a great framework to try and understand sex differences while also thinking about it in this context of brain and behavior, and then highlight some of the work that we've done. So in terms of these analyses looking at differences across males and females, traditional studies have primarily looked at group differences. So how is the average male different from the average female? But with increased access to big data and computational resources, we can actually use what we call brain-based predictive modeling approaches to understand how neurobiology is related to behavior at a group level, and then use that to make predictions at an individual level. And these lead to different types of analyses that are called descriptive, predictive, and prescriptive. And I'm not going to go into too much detail here about them, but these are all different types of machine learning analyses that can yield clinically meaningful insights that we can then use to develop personalized diagnostic tools and therapeutic interventions. As an example, in the context of substance use and addiction, descriptive analyses can be used to identify biological subtypes of addiction or substance use disorders. Predictive analyses can be used to predict how vulnerable someone might be to develop an addiction. And prescriptive analyses can help us understand how likely someone is to respond to an actual intervention or a treatment. And when we think about these types of analyses and the brain-based predictive models that we use to conduct them, we have to recognize that there are a lot of different choices that we make in the modeling or the mathematical process that can affect model performance and interpretability. These choices exist in terms of the aspects of the brain modality and state that we choose to base our predictions on, the parcellations that we use to segment the data, how we transform our data before we actually put it into the model, the sample size that we have, whether we use population-specific or general models, the algorithms we're relying on, and the phenotypes that we're looking to predict. And today in particular, I'm going to be talking about the use of population-specific models because they provide a simple yet very powerful framework that we can leverage to understand potential sex differences in brain behavior relationships. So basically, when we're developing these machine learning models, we can either choose to base them on the entire sample, or we can split the sample into specific groups based on some criteria like sex. Then we can actually model the relationships in each sex separately and compare how similar and different they are. And not only that, we can also see if models that were captured in males generalize to predict the same things in females and vice versa. So it's a great way to understand and quantify multivariate sex differences in the brain, which actually take into account the whole brain rather than just looking at one aspect of the brain at a time. So in the last couple of minutes, I'll just briefly talk about a couple of examples of these types of studies, and then I'll wrap up. So in the types of analyses that we do, we primarily use functional MRI data, and functional MRI measures brain activity by detecting changes in blood flow coupled with neuronal activation. And these functional activation signals can actually be used to compute what we call functional connectivity, which tells us how similarly or how correlated two brain regions are in their activity, or how they are co-activated over time. And these functional connections are different across the sexes. So in recent work, we've looked at how they vary and the types of functional connections that are actually associated with sex. And what we found is that connections within and between control, limbic, attention, somatomotor, and visual networks are highly important for this. So in other words, in a multivariate sense, sex differences are not limited to just a single region or a small set of regions. They are actually widespread throughout the brain. And it's not just sex that's influencing these networks. We actually also showed that gender, independently of sex, is further influencing this. And a lot of times, because we don't try to actually parse out sex and gender in our studies, this further tells us that gender might be influencing what we perceive to be sex differences. So specifically here, we were looking at the functional connections that were associated with gender in assigned females at birth shown on the left and assigned males at birth shown on the right. And what we found is that gender primarily influences temporal parietal, default, and attention networks in females, and temporal parietal, default, attention, limbic, and visual networks in males. So not only are these brain networks associated with sex and gender, that association with gender might actually also be slightly different across the sexes. And in terms of behaviors, we tend to look at a lot of behaviors that are linked to different mental health conditions. So in this study, we were specifically looking at externalizing behaviors, which represent behaviors that are directed at others or the environment. So this is things like rule breaking and aggression, and internalizing behaviors, which are directed at oneself and include things like anxiety, depression, and somatic complaints. So in these analyses, what we actually found is that the same set of brain networks and functional connections that underlie externalizing behaviors in males, shown on the left, actually predict internalizing behaviors in females, shown on the right, and vice versa. So this tells us that the same set of functional connections that might be representing mental distress in the brain can manifest as different behaviors across the sexes. So there might be some aspect of sex and gender that are uniquely interacting across these different scenarios to lead to different behaviors, even when the brain patterns underlying them are the same. And finally, we're recently also started looking at how these connections differ across individuals with psychotic-like experiences. And here we found that in males, psychotic experiences primarily map onto functional connections in limbic, somatomotor, and visual networks, and this is shown on the right. Whereas in females, they're kind of more dispersed throughout the brain. So unlike externalizing and internalizing behaviors, psychotic-like experiences actually map onto distinct brain networks across the sexes. So to briefly summarize, sex differences may exist in brain behavior or brain behavior relationships. And these sex-specific brain-based predictive models enable us to examine how sex influences these relationships in neuroscience and psychiatry. Then we have a few summaries, highlights from the studies, which I won't go into. And with that, I just want to thank my entire lab, my collaborators that contributed to the work that was presented, and the opportunity to present here. Thank you.
Thank you so much. That was great. And our next speaker is Erin Capilari from, she's an associate professor at Vanderbilt University. Erin, pass it over to you.
Hi. Okay, you think I'd be good at sharing my screen after all of that nonsense, but it still takes me a minute. Okay, I think we're good. I'm really excited to talk about this project. This is actually new work for my lab, focusing on understanding how hormones act in the brain and how this could potentially influence the way that the same circuits work in males and females. The dopamine system is my favorite neuromodulatory system. I know lots of you all study this as well. And, you know, when we think about dopamine, it's really at the hub of studying female vulnerability and addiction, because nearly all drugs of abuse increase dopamine levels in reward-related brain regions. This dopamine signal is really critical for learned behavior and reinforcement. But when we think about dopamine release in these regions, we really often think about kind of cell body firing in the ventral tegmental area leading to increases in release. But there's also a lot of regulation that happens at distal axon terminals in the nucleus accumbens that can happen through all of these different modulatory systems. It's really often overlooked, and so don't worry, there won't be a test. But the one that I most often study, or have been studying in my lab for the past few years, is really the regulation that's happening through nicotinic acetylcholine receptors located on dopamine axons in the nucleus accumbens. So these receptors are really important for a whole host of reasons. First, they're the primary target for nicotine itself. So obviously any differences in these receptors has really broad implications for addiction. But they also are really key regulators of when and how much dopamine is released. And so these receptors are made up, they're pentameric receptors made up of the different subunit combinations. So they can be homomeric or heteromeric. But the combination that is most often seen in the nucleus accumbens are these alpha-4 beta-2 containing nicotinic receptors. So I'm going to talk about the receptor generally, but the subunit specificity will come back at the end. So it's a little foreshadowing for you all. So, you know, what do these receptors do? So they're actually located on these dopamine axons, and acetylcholine is released locally from cholinergic interneurons and acts on these receptors, and this actually leads to regulation of dopamine release. And so, we'll talk a little bit in a minute about the frequency mode of dopamine firing, but essentially in a lot of situations, it can potentiate how much dopamine release is happening. These receptors are also really interesting. They pass sodium and calcium. These are the major ions that are passed through them. But they also have a whole host of different sites that can actually regulate their biophysical properties, which actually I'll give away the ending. What I'm going to show you is that estradiol actually interacts with these receptors directly to change how they work and alter dopamine release between males and females. So they are really interesting receptors. They don't just ubiquitously increase release. People think of these, they talk about them as a low pass filter. And so what happens is when you actually antagonize these receptors or you block them, you get reductions in low frequency release. So if dopamine is firing at tonic modes, these are just like background firing rates. You'll get decreases in release that's happening at those modes. But if there are bursting events, like we call these aphasic events that happen, like for example, in response to salient stimuli, if you remove these nicotinic receptors, this is greatly potentiated. And so this is kind of that like textbook mechanism that we talk about of dopamine release regulation. And in my lab some years ago, Lillian Brady started trying to understand how there could potentially be sex differences in dopamine regulation. And what she found was that in intact females, this regulatory mechanism does not seem to be present. And so this is obviously really important for a whole number of reasons, but because it's guiding a lot of experimentation in the field, it's really important to know that this is kind of a fundamental difference in how the system can be regulated. So, we kind of went on to say, well, why is this happening? There's a whole number of reasons this could be happening. But we started first by looking at hormonal regulation. And so one of the questions is this an organizational difference where these receptors are just not there in females? Or is there maybe something happening in adulthood that's kind of changing their relationship? And so what she did was she overreactimized females. And what she found was that this regulation by nicotinic receptors could be completely rescued. So this shows you two things. One, hormonal factors in adulthood are involved. But two, nicotinic receptors are present and functional in females under some conditions. So, I told you that we're going to look at different actions of nicotinic estradiol and nicotinic receptors. So this is some work from oocytes. And what they were doing in this study is they were cleverly expressing different subunit combinations of these nicotinic receptors. And so what you have here on the left is the alpha-4 beta-2. This is the receptor combination that's on those dopamine axons. And what you can see is they're measuring currents passed through these receptors. This is in the presence of 1 micromolar acetylcholine. What they did next is they added estradiol to these cells and they redid this acetylcholine experiment. And what you can see right off the bat is what's happening is that estradiol is potentiating currents through these receptors. So if you look at the kind of spread of different subunit combinations, the one that is most affected by estradiol in these studies are those combinations of receptors that are located directly on dopamine axons. And so the question became to us is estradiol having some of its actions on dopamine release in the nucleus accumbens through actions on these alpha-4 beta-2 containing nicotinic receptors? And so what our hypothesis is that estradiol acutely actually potentiates the function of these receptors. But these receptors are known to be very highly sensitive to desensitization. And so what I think happens is in intact adult females, this potentiation leads to desensitization, which essentially reduces the modulatory effect of this receptor on dopamine release over longer periods of time. So I'll show you, I don't have I have 10 minutes. So I'm going to show you some of the key findings, but not all of them. But the three things that you need to know first off is really what do we need to know to show that estradiol is acting through these mechanisms? One, estradiol should potentiate dopamine release. The effect should be larger in males than females because that effect is likely driven by these receptors. And we should be able to completely block the rapid actions of estradiol by blocking these receptors in the presence of estradiol. So what this is a study, each dot is a time. So we're recording dopamine release. This is actually with voltammetry. And what you can see in this like peach color is that's when we add estradiol to the slice. And what you can see in these white boxes is that estradiol increases dopamine release, and it does it very quickly. What's even more interesting is that if you do this in the presence of a nicotinic receptor antagonist, DHbeta-E, it blocks the ability of estradiol to increase dopamine release under these conditions. In intact females, what you can see is there is not an effect any longer of DHbeta-E, that nicotinic antagonist, but we can completely rescue this effect again with ovariectomy. And so there's an interaction here between these effects and the hormone state of the animal. But what's really important is that these rapid effects of 17-beta estradiol on dopamine release are through a nicotinic receptor mechanism. I'll tell you some of what we know, but I can't show you it all. Estradiol rapidly potentiates dopamine release. This is happening very quickly. We can block it by blocking these nicotinic receptors. This actually leads to reduced effects of both antagonism, so this DHbeta-E effect I'm showing you, but also agonism. So we did a bunch of experiments, and I'll show you some at the end, with nicotine and neostigmine, and it's the receptor is hypofunctional in both directions. It's specific to these alpha-4 beta-2 containing receptors. We did a bunch of experiments with alpha-6 and alpha-7 subunits that did not have any effect. And we can't block the effects of estradiol on this timescale by estrogen receptor antagonists. And so this effect seems to be very specific to these, what we think are direct effects on nicotinic receptors.
Erin, can you kind of conclude it up or tie it up?
Yeah, sorry, I thought I had 10 minutes, and it's been eight minutes.
Yeah.
Okay, yeah. Basically what I'll end with is we did a bunch of structural biology, and we found two potential estradiol binding sites that are at this alpha-4 beta-2 interface. And so these seem to be really important sites for this regulation. And this site actually overlaps with known agonist sites. And so essentially what I'll end with is that nicotinic receptor function is altered in females in a way that really alters how they regulate dopamine release. These effects seem to be mediated by hormones and change the way that the same circuit is regulated between males and females. And what we think we've really identified as a way that hormones can act in the brain independently of these traditional estrogen receptor signaling mechanisms to change how the brain controls behavior. Okay, with that, I'll thank everybody, and thank you.
Great. Thank you so much, Erin. And now we'll move on to Deena Walker, who's an associate professor at Oregon Health and Science University.
Thank you. Can everyone hear me and
Yes.
see my screen? Okay, great.
Right.
Oops, sorry, just a second. Wait. Okay, so I'm really excited to be here, and I want to thank the organizers for inviting me. And my talk today will focus on sex differences in transcription and gene regulation. So it's good that I followed the two previous presenters, because it gives an example of how drugs of abuse impact dopamine. But what we also know is that drugs of abuse then impact transcription through these second messenger cascades. So you have your neuron here that releases neurotransmitter, and then that then leads to second messenger cascades, which goes into the nucleus, leads to transcription factors binding the DNA and increasing RNA levels, and then that leads to translation and the production of proteins. And we know that transcription can be changed after one dose of cocaine or one dose of a drug, but it can also, repeated exposures to drugs of abuse can change transcription, mostly through epigenetic mechanisms. So this is some work by Philip Muse showing that repeated exposure of cocaine can induce differentially open chromatin. I'm not going to go into this super deeply, but just know that the more red there is, that means there's more open chromatin, which means there's more likely to turn on transcription. And then recently, Philip expanded on this to show that repeated exposures to cocaine also, this seems to happen through persistently altering histone marks and even histone composition. And this is a really detailed graph, and I don't really want you to go look too much at it, but I wanted to show you that even then you can then change the makeup of the histones within there after repeated exposures. So this is important because what we know is that at all of these different mechanisms of gene regulation and transcription, there are sex differences in this. This is partly due to the fact that hormones, when they bind their receptors, are in fact transcription factors. So they interact with the DNA and change the makeup of the RNA and proteins within a cell. So I've been interested in how cocaine influences transcription for a while, and one of the first studies I did was to look at how transcription was impacted by drugs of abuse with one injection, and we found very quickly that there's very little overlap in the transcriptional signatures in response to one dose of cocaine. So this is just looking at a heat map on top here. The males are on the top, the females are on the bottom. Genes that are downregulated are in blue, and genes that are upregulated are in yellow. And what I hope you can see here is that from the Venn diagrams as well as the heat maps, that there's a lot of genes that are regulated in males, shown in blue, and fewer genes regulated by one dose of cocaine in females. And this was similar across the 3 brain regions we looked at. And then when we dug into this and looked to see what the upstream regulators were, we found that there were predicted upstream regulators. We found that there were differences in those as well. And I just want to point out one finding that we had here, which was that while dopamine was a predicted upstream regulator in the male data, it didn't come up in any of the brain regions in the females. And I think you can convince me of a lot of things, but I don't think that the cocaine pharmacology doesn't involve dopamine signaling in females. I think it's important here to point out that most of these upstream regulator databases are based on previously published work, and almost all of that has been published in males. So when you have bias in the data sets that make up these databases, you end up with bias in the downstream results that you look at here. So it wasn't just the reward circuitry that we saw these differences in cocaine. We looked at a brain region called the medial amygdala, which is a known sex-specific brain region, and we saw similar effects. So this is looking at 1 dose of cocaine euthanized 1 hour later, or 24 or 10 doses of cocaine euthanized 24 hours later. And similarly, there are a lot more genes regulated by the first dose of cocaine and by chronic cocaine in the males on the top than in the females on the bottom. But we took this step a little further, and we did some more bioinformatic analysis to see if we could identify genes that were changing together using co-expression analysis. And when we did this, we identified different gene modules that showed sex differences in their connectivity and hubs within those gene modules that might be important. And one of the ones that we found that was pretty striking was this gene module that had the hub gene CRIM, crystalline mu. And you can see here on the top, the gene module that includes CRIM has CRIM as very central to the module. It has a lot of connections. It's a large hub gene, whereas in the females, it's in the periphery of the module. It has fewer connections. It's less of a hub gene. And we hypothesized that if we manipulated this gene, that we might see differences in cocaine-related behaviors. So we then we overexpressed crystalline mu into the medial amygdala in adult animals, and we did see that when you overexpress crystalline mu, it upregulates, or it enhances cocaine CPP in males, but not females. And then it has other effects on anxiety-related behaviors. And because crystalline mu is a thyroid hormone binding protein, and as I mentioned, thyroid hormone receptors are transcription factors, we thought that we might see differences in transcription when we overexpress this gene. And sure enough, that's what we found. So on the top, we see the number of genes that are regulated by overexpressing crystalline mu in this sex-specific brain region. On the bottom, you see that there's a lot fewer genes that are regulated in females. And the other thing I want to point out here is that there's a lot more genes that are downregulated, shown in blue here. And so this told us that the medial amygdala might be a critical region for sex differences in reward-related behaviors. And because of its role in thyroid hormone binding, we thought that thyroid hormone signaling in the medial amygdala might be important in mediating some of these sex-specific behaviors that we saw. So just to summarize, we saw that when we overexpress crystalline mu, this thyroid hormone binding protein, we see an increase in transcriptional repression and an increase in the preference of cocaine, but only in males. And so we did a little digging into what thyroid hormone does and how thyroid hormone regulates gene expression. And what we found is that there's differences in how you get activation and repression of these genes depending on the receptor that it binds. So when the ligand is available, the ligand is thyroid hormone, it binds its receptors, the receptors homodimerize, go to the DNA, they bind the DNA at these response elements called thyroid hormone response elements, and they activate transcription. But when there's thyroid hormone is unavailable, like when you overexpress the binding protein and sequester thyroid hormone from the nucleus, thyroid hormone receptors are more likely to engage in ligand-independent transcriptional repression. So this means thyroid hormone receptor binds with another receptor, it moves to the DNA, and it recruits a bunch of repressive proteins to shut down transcription. And thyroid hormone receptor alpha is more likely than thyroid hormone receptor beta to engage in this. And so we wondered if there were differences in the expression levels of thyroid hormone receptor beta in the medial amygdala that might explain some of this. So we just took some animals and we did basic qPCR, and indeed that's what we found. We saw females have higher levels of thyroid hormone receptor beta than males in the medial amygdala. And we started wondering if maybe this level of thyroid hormone receptor beta was protective for some of these effects that we were seeing in the animals. So our hypothesis was that thyroid hormone receptor beta mediated transcription was protective in these females against this thyroid hormone dysregulation. And we figured that if you upregulate THR beta, that you could decrease cocaine reward. But if you have lower levels of THR beta, you have increase in ligand-independent repression, and that leads to an increase in cocaine reward. And so we were ready to test this hypothesis. I was super excited. I had a new grad student come in. And then my grad student said, I'd really like to look at alcohol instead. And so instead of looking at how cocaine reward was impacted by thyroid hormone receptor beta, we looked at alcohol. And in this experiment, we worked with our collaborator, Dr. Tom Scanlon here at OHSU, to answer the question if activating thyroid hormone receptor beta within this CNS can affect alcohol drinking. So he's developed this CNS-specific, THR beta-specific agonist called SOB-AM2. It's a prodrug. You inject it into the animals, it gets transported in the brain, converted into its active form, and then acts at thyroid hormone receptor beta specifically. And we just did a very simple 2-bottle choice test and waited until the animals had established drinking, shown here, and started injecting them with this prodrug called SOB-AM2. And so this is the results of the drinking study there. The first is showing the males in the darker colors, the females in the lighter colors. And you can see that the established drinking patterns were not different across the groups. When we treated with a low dose of SOB-AM2, we didn't see much of an effect on drinking behavior. But when we upped the dose to 1 mg per kg, we found that especially in the males shown in red, SOB-AM2 suppressed their drinking and their preference for cocaine, or their preference for ethanol, sorry. We saw, when we averaged the data, we saw similar effects in the females, but the higher dose reduced their intake and their preference, but it wasn't significantly different from the vehicle-treated animals. And we actually think this is a difference in the way the vehicle-treated animals are drinking, less so than the way that the SOB-AM2, but we're repeating this right now to see if that really holds up. But what we can take from this is that first, drugs of abuse persistently alter transcription, likely through epigenetic processes, and these processes are sex-specific and brain region-specific as well. That there's sex differences in transcription, in the transcriptional response to cocaine, and they're observed throughout the reward circuitry. That when we integrate multiple bioinformatic approaches, we can identify sex-specific regulators of cocaine-related reward and transcription, and maybe even alcohol-related reward and transcription. And that by leveraging some of these sex differences that we see in the response to drugs of abuse, This will now enables us to identify treatments for substance and alcohol use disorder for all vulnerable populations. And with that, I'd like to thank the current members of my lab, particularly Michael Johnson. He's the one who really took that SOBAM2 experiment and ran with it. The past members of my lab were instrumental in helping me get set up, and the collaborators for these experiments are Tom Scanlon and Dr. Andrea Binion.
Thank you. Thank you, Deena. Thank you to all the speakers. There was great talks. We have about 5 minutes. We were a little over, so I want to get us back on track. So we have about 5 minutes of questions, and I'm going to take the liberty to ask the first. They're all great talks, and I have a lot of questions, but I'm going to take the liberty to ask the first question, and it's to Erin Capilari. Your sex difference kind of focused on hormones, and so it makes me think of changes in the vulnerability to addiction across the lifespan. So can you comment on that? Oh, Erin, I think, are you muted?
Oh, sorry, I was muted. Two ways. I actually think this is one of the most interesting aspects of this data, is that it provides a mechanism where you could have factors that are there and really functional at specific developmental periods. The question becomes, though, whether hypofunctional those receptors being hypofunctional
Hi, Erin. I think that you've frozen, so we might take another question if that's okay, Rita.
Yeah, I'm sorry. I thought I froze. I was trying to fix it.
No
Sorry.
problem.
So I think Hugh had a question, and I believe this is for Deena. Other than that, it works and shows effects. What's the rationale for looking for gene expression effects after one cocaine administration?
Oh, yeah, the reason we look at both, I didn't show the chronic data because I only had 10 minutes. But the reason we looked at acute and 10 doses of cocaine was we wanted to know the immediate transcriptional effects and the immediate transcriptional profile, because we assumed that it would be different between males and females. And you're not going to get that. We wanted to know if the transcriptional profile sort of changed over time. And so we needed that first baseline measurement to determine what happened after repeated exposures.
Do those effects persist for an amount of time after a single exposure?
So it's kind of amazing that doesn't seem to matter whether they're the first dose of cocaine or whether they get repeated exposures. There's always really big differences in the way that males and females respond transcriptionally. If you look in human data, I didn't go into this because I was at a time crunch, but if you look at the human data, there's very little overlap in sex differences in human data. There's very little overlap in mice. There's very little overlap in rats. It is just really, really different. I have many ideas of why that might be, but I don't know if you want to sit around and chat about that right now.
Right, that's great. Thank you.